Translation is how the information in genes gets expressed. In other words, translation is how the information carried by genes on the chromosomes is used to make proteins. Tying together transcription and translation, genetic code written in DNA is transcribed into messenger RNA. Ribosomes use messenger RNA, transfer RNA, and amino acids to translate the genetic information into specific polypeptides. The horizontal line, beginning with start and ending with stop, represents a messenger RNA molecule that has left the nucleus and is now out in the cytoplasm. The information on the messenger RNA is written in the form of sequences of 3 nucleotides called codons. The start and stop codons tell the ribosome where to begin and end. The other codons specify a specific amino acid. In my example above, for the sake of simplicity, I've made each codon have 3 identical nucleotides. While this is possible, codons can also consist of any combination of A, U, C, or G. The sequence was determined by the nucleotide sequence on the DNA that was used to make the messenger RNA. Translation begins with a ribosome attaching to the first 2 codons. The codon that means start, and, in my example here, the second codon, AAA. Transfer RNA next arrives. There are several different transfer RNA molecules. Each different type carries a specific amino acid and has a specific 3 nucleotide sequence called the anticodon. Similar to what we saw during transcription, A will form hydrogen bonds with U, and C will only bond with G. Since the codon on my messenger RNA is AAA, only the transfer RNA molecule with the anticodon UUU will attach to the messenger RNA. This specific transfer RNA molecule carries only one particular amino acid, which I've called AA number 1. With the arrival of the blue transfer RNA molecule and its amino acid, the ribosome now moves over 1 codon. The ribosome is still covering the codon AAA and now also the codon CCC. A second transfer RNA molecule arrives, shown here in green. This transfer RNA molecule must have the anticodon GGG. It brings with it a different amino acid, in my example, AA number 2. The ribosome now catalyzes the formation of a peptide bond between the 2 amino acids. After the formation of the peptide bond, the first transfer RNA molecule is released, leaving behind its amino acid. The transfer RNA molecule will now go find another molecule of amino acid number 1. The ribosome moves over 1 codon. The third transfer RNA molecule arrives with its amino acid. Codon and anticodon have to complement, so the transfer RNA molecule can attach to the ribosome messenger RNA complex. A new peptide bond is formed between the amino acids number 2 and number 3. And then our second transfer RNA is released. The ribosome moves over 1 codon. The transfer RNA with the appropriate anticodon arrives. A peptide bond is formed, and the previous transfer RNA is released. This process continues down the length of the messenger RNA molecule until the ribosome encounters the stop codon. The last transfer RNA is released, and also released is the completed polypeptide. The completed polypeptide has been released from the ribosome and the messenger RNA. Notice the messenger RNA has not been changed in any way. If more of this polypeptide is needed, a ribosome can attach at the other end at the start, and multiple copies of the polypeptide can be produced. If no more of the polypeptide is needed, then the messenger RNA can be disassembled and individual nucleotides can be used to make more RNA. So, translation uses ribosomes, messenger RNA, transfer RNA, and amino acids to make polypeptides.